going to look at the principles of serology, and I want us to look at the introduction. What is this serology that we are talking about? Because this is what you probably be engaged in trying to understand the diagnosis of diseases as it is applicable in hospital or clinical setup. So let's begin our discussion by looking at the learning objectives that you need to know by the end of the discussion today and tomorrow. So you need to understand the following. You need to be aware of the laboratory safety rules, or rather the guidelines that govern the laboratory practice or laboratory method. These are very critical. Even though you are a usual member, you have to adhere to these guidelines for the safety of yourself and others. Then you also need to list different types of serological tests that are used, state the principle of serological tests, describe the application of different serological techniques. When do you need a particular serological test, and why is it important? As you are aware, sometimes we have these new infection or emerging infections. People are talking about using the molecular techniques. Why not antibody techniques? And if you use, would they make sense? For example, somebody is vaccinated. So that's why the choice of these techniques in different applications is very, very critical. Then also you need to identify the advantages and the drawbacks of different serological tests that are in use. No one test is 100% accurate or applicable to every diagnosis. There are some limitations and advantages, as we discover later on. Then you also need to identify some of the factors that affect the antigen-antibody interaction. Why should you consider these factors even before you draw your conclusions? It's very, very critical, especially when you know about immunology. There is even close reactivity. There are some aspects of the boson effect, that is having at least equal amount or concentration of antigen and antibody. And in that case, you need to use some dilutions where applicable. In some situations, you don't need to dilute if this has been done by the manufacturer at the time of the manufacturing process. So these are some of the things that each and every reagent kit or immunological test has the instructions that have to be followed. If they are not there, then they must be shared in terms of standard operating procedures, which will ensure that everybody does the same thing in the same way. Now, overall, let's look at the general laboratory safety, because when you talk about serological tests, these are the tests that has to be done in a laboratory environment. And for that reason, there are some guidelines that are very, very critical, like I said earlier on, that need to be adhered to. So this constitutes what we sum it as laboratory safety. Now, all students must read and understand the information of the laboratory safety and emergency procedures prior to the laboratory session. So if you are entering any lab, be it a clinical or a diagnostic, sometimes even a research, clinical research lab, one thing is to understand or request for these guidelines that guides these personnel that are working in that area. For example, if you go to radiology, you must be seeing different attire. If you go to level 3 laboratory, you'll see different attire. And this tells you the level of exposure which you must adhere to before you engage yourself or you touch anything. So that's why that this statement says that your personal laboratory safety depends mostly on you. Sometimes people may not tell you, but you have to be inquisitive and ask, because it's about your safety. Now, we look at briefly some of this safety we are talking about. We have those general or the universal laboratory safety procedures which we look at. There is also the electrical safety. There is the chemical, those who are preparing reagents. There is also biological. How do you dispose the waste, especially when it's infectious? And emergency response in the case of a spill in the laboratory or in case of an acid how you're supposed to deal with all these emergency responses, because accidents are bound to happen. So these are some of the things that people need to have um, um, to be aware of before they start or they engage themselves. Now, the general laboratory procedures, as the figure shows you that there is danger, and for that reason, you need to be cognizant of these safety rules if you have to be safe. Number one, as a normal practice, each and every person needs to wear a lab coat. And this lab coat is just one of the protective gear that everybody is expected. So whenever you go to a lab, always make sure you have one laptop and it is properly fitted. If it has buttons, it must be well kept and all that. Never eat or drink in the laboratory. I've seen sometimes people are chewing in the lab, and this is not acceptable at all. Read labels carefully. You know, people will be dealing with very dangerous chemicals, and sometimes somebody may forget. So in that case, you need to ask, is this an acid or is it water? Because everything looks the same under glass or through a glass. So you have to be careful, because labeling is very important. So if you're the one working, you make sure everything is well labeled and kept to avoid any accident. You also need to wear safety glasses and face shields where applicable. And this is just an additional, especially those who are working in highly infectious environment like level 3 and sometimes level 2 labs that have been modified. You also need to wear gloves when using any hazardous or toxic agent, just to avoid contamination with your skin. Sometimes it may cause serious irritation. Clothing, that is, sandals should not be worn in the laboratory at any time. Shoes are required. So if you work in a laboratory, there are some attire in terms of the clothing that you need to consider so that you are safe in that laboratory. So there are many standards we may go through, but just these are the few ones that I want to highlight. Then keep the work area clean of all materials, especially whenever you go to a lab, you're not the one who was working there. Somebody else was working. So first is to disinfect or make the place clean for any work to be done. So this is a normal practice before you start any work, and after you finish school, the place has to be cleaned. Extra books, puzzles, or its personal belongings should be kept away from equipment, especially when you deal with highly contagious material. And sometimes because you don't know any sample that it comes, so and the people touch here and there, so you have to keep the personal belongings on a totally different place. Students are responsible for the proper disposal of used material, if any, in appropriate containers. So you are responsible for whatever you use. If you are in a practical, ask how do you dispose of or what procedure is involved so that you don't leave anything at risk. Equipment failure, in case there is one, this needs to be reported immediately so that it can be either be serviced, or because it is performing critical assays, I guess. Because if a patient provides a sample, there is no results, you can imagine what will follow thereafter. And we don't want to delay, that is causing more anxiety. So this equipment that is malfunctioning or is not working has to be reported, and never try to fix it yourself, because you could harm yourself if you don't know how it works. So there are professionals who have been trained and who can help. What you can fix is small things. Yes, you can troubleshoot, but not working an area that you are not familiar. Now, clean up your work area before leaving. As I said earlier on, this is a practice before and after you do your work. Then you also need to be aware that your hands needs to be washed before leaving the lab and before eating. So that those are the universal safety precautions that you expect to govern any lab around the world, not only in Kenya. Now, the emergency response, it is your responsibility to read safety and fire alarm posters wherever they are displayed, and this will be a very important safety procedure for any person. So whenever you are working, always look around, see what is uh, labeled as uh, dangerous or biohazardous, and these are some of the things that you just see by your naked eyes by looking around and seeing what happens. The fire alarms are always there, and there is always a key which is hidden somewhere, and what you need is to break the glass, and there you just activate the alarm. But there has to be a training. You don't have to do it if you have never done it before. You know, you also need to know the location of the fire extinguishers, the eye wash in the case of a lab spill or something that has happened. Then a safety shower. Of course, we are talking about advanced areas, especially where you'll be working all the time. In the environment that you might be involved, you may not see everything there, but it depends on the level. You know, there's level 1, level 2, level 3 labs. So each lab has different safety requirements. Notify your instructor immediately after any injury or a fire or explosion or spill. Then lastly, you know, the building evacuation procedures whenever you are working. Yeah. Now let's look briefly about terminology. Having talked about the safety procedures as you enter in lab, then you are ready to start looking at some reactions or serological reactions in the lab. But first, what happens in the body system that leads to us or to you performing any serological tests? Now, as you are aware that the immune system is constituted of the cells and also the solvable constituents that allow the host to recognize and respond to foreign stimulants. For example, we talk about the immune cells, the white blood cells, the T cells, the B cells, the innate immune cells, monocytes, macrophages, and so forth. These are all organized in such a way that there is always the first line of defense, there's the second line of defense, and there's the third line of defense. Now, the first line is usually the physical barriers, your skin, your secretions, the pH, and all that. So these are just the first line of defense is just to protect, especially mechanical barriers. But now, when we talk about the second line of immunity, now we talk about the immune cells, and specifically, it begins with the innate immune cells, where you find the antigens or the foreign particles are recognized, and then the innate immune cells leading to the second, the third line of defense, which is now the specific arm of immunity that is composed of the T cells and the B cells. Now, in terms of the primary immune response, we talk about the physical barriers. Now, the secondary immune responses, and that is what I'm going to concentrate specifically, like we talk about the immune cells. Now, these immune cells, I've said we have the innate immune cells and we have the specific immune cells. Now, innate, we talk about the basic immune cells that identified antigens, the macrophages, the dendritic cells. These are cells that can phagocytose, or they can engulf any foreign particles, but presents it to the second, to the specific arm of immunity. Now, that is the B cells. Now, at this time, when the antigen interacts with the immune cells, now what happens is that the immune has to be activated. Now, when it is activated, if it's the T cells, they have to differentiate into memory T cells. If it's a B cells, they have to activate into plasma cell and differentiate into finally the antibodies or antibody molecules. Now, these are some of the basic expectations of any interaction between the immune system and an antigen. And in that way, this process is happening in the natural environment or in the host. So what happens next? Now, these molecules, for example, the antibodies that have been generated by the activation of B cells, they can interact with antigens. And if we bring this interaction in vitro or in the laboratory environment, this process can be reversible. In other words, the antigen can bind with an antigen, and sometimes if you vary the concentrations, you can have the reverse. The antigens can dissociate from an antigen and so forth. So these antigen-antibody interactions are very, very critical because it will enable us to detect and measure these reactions, which forms the basis of serology. So some people talk about serology as the study of antibodies, but in the modern way, you study antibodies in combination or when you react it with an antigen. So an antibody cannot react on its own unless there is an antigen component. So therefore, serology is the science that deals with the measurement of an antibody or an antigen in fluids. So, or in solution form, that is. So you can have your antigen source and your antibody source. So sometimes we have these reagents that are prepared commercially. Some have already immobilized antibodies within themselves or immobilized antigen. So whichever that is applicable, it is immobilized so that it can be used to capture the other component. Now, then what is serology? It's a branch of immunology that deals with the study of antigen-antibody interaction. And I've said this normally happens in vitro or in the laboratory setup by different serological tests. So we have different techniques, and this one we study them in detail tomorrow. What are these immunological techniques that are used, especially in laboratory? We will look at them one by one, the most common ones, the application, and so forth. So for today, let's look at the basic or the principle behind this serological test. Now, when you have this antigen-antibody interaction, it is possible that you can detect or you can diagnose the presence of either an antigen or an antibody using the samples that you can obtain from the patient. So what is then the importance of this serological test? Number one, they are very critical in saving the patient's life. If you don't know what is the cause of the infection, you cannot know the right treatment to give for this particular patient. So by knowing what is the cause of the disease, you can formulate the best or the appropriate treatment. Then it's also very important to prevent the spread of disease. For example, if somebody is known to have COVID, for example, that is now in the limelight, then you are either advised to isolate yourself so that you don't continue transmitting or spreading to other people. Then it's also very critical for treatment therapy because we have different treatments available for each and every disease, and that is very critical. And that can only be done when you have found that this person is infected by looking at the presence of an antigen or an antibody. And then finally, you can confirm the clinical diagnosis. Usually, the diagnosis can be confirmed by antigen detection because antibody detection alone may not give you the confirmation. The reason is somebody who is having natural infection may have recovered, but still has the antibodies to that particular infection. So, for example, somebody who is also vaccinated, you can have antibodies because these are mimicking the antigen that is causing that particular disease. So looking at the antibodies, it tells you somebody is having immune response to a particular antigen, but not necessarily saying somebody is still having an active infection. This can only be done by either looking only one specific immunoglobulin, that is immunoglobulin IgM, which is very critical in differentiating between a reinfection. For that reason, when somebody is reinfected again, because the titers will tend to rise. But with immunoglobulin G, it only tells you there is immune response against this particular antigen, and that is memory that is stored. Okay. Then laboratory diagnosis of infectious diseases. Number one, let's look at the isolation and identification of the causative agent. The common norm is that people look at morphological tests, and that is looking at them under microscope, look at the features that are very particular to that particular. If it's a bacterium, you can even see it on a slide, as long as you use the correct staining procedures, like gram stain. If you're looking at gram negative or gram positive, you can check against the background. Then you can also use the chemical reactions. For example, you can check the oxidase test. You can check the lactose fermenting organisms. Those are just biochemical tests that you can learn much more about this in microbiology. Then you can also use cultural identification of these pathogens, meaning you need to culture them, and you can study the cytopathic effects, maybe by looking at the morphology, the size, and some lysing properties of this particular pathogens, if a virus that you want to look at the lysing or infection of the culture cells and so forth. And this is done in a tissue culture laboratory. Then you can also now check the serological reactions, and that is our interest, by looking at the antigens or the antibodies to a particular antigen that are produced. And nowadays, there's the utilization of the biotechnology whereby you use the PCR, that is nucleic acid tests, the DNA and the RNA techniques. All these are available beyond what you just see on the slide. So there are many molecular techniques currently available for detection of these pathogens. So in terms of detection of specific antibodies in sera of infected patient, using the techniques, there are specific kits that you find available. For example, if I need to detect antibodies against HIV, I look at different HIV kits that are in the market. And of course, some of these kits must be approved by a renowned body. For example, the Food and the Drugs Agency, FDA from US, or the WHO. These are commonly used as the rubber stamp, just to make sure that the kit has passed the manufacturing process and it meets the requirements of standard, so that it can give almost accurate results. If this is not achieved, then these kits cannot be licensed. And that's why, for example, getting the COVID-19 antibody test, until now, there's no really kind of a consensus on antibody detection kits because of the challenges with this antibody test and the changing nature of the COVID-19 or the SARS-CoV-2. So for that reason, you must have this approved for any kit that is in the market. Of course, there are many kits out there that are used for detecting, but they are not used for laboratory diagnostic, and they are only used for research. So have this difference that whenever you find some kits, maybe in a research environment, don't expect to find the same kit in a diagnostic laboratory or in a clinical laboratory. But sometimes you may find the same kit, if it is well approved, you can find it either way in lab or in research setup. Then diagnosis of other diseases or change of condition. So these are some of the applications that you can utilize in terms of diagnosing for these infectious pathogens. Now let's look at the serological reactions. The primary reaction deals with the interaction between an antigen and an antibody in vitro, that is inside the test tube. So you can have your test tube, either the antigen, and introduce the antibody and allow it to react, so you can be able to see what happens. So in this case, you have examples like ELISA, IFA, that is immunofluorescent antigen test. You can talk about the radioimmunoassay tests, and then you talk about enzyme-linked immunosorbent assay, that is ELISA. So some of the examples, and then we look at them tomorrow. Now, why do we say it's primary? Because you just reacting direct, so if you have your antibody from the sample, then the tube, you introduce an antigen, you expect a reaction to be seen, and now you can be sure that you are doing the right thing, or you are getting some positive or negative results, or whatever situation. Then secondary, that is number 2, this measures the consequence of interaction between antigen and an antibody. For example, when you have agglutination tells you there is an antibody and there is an antigen, and that comes into something that is visible in terms of flocculation or agglutination. And these are the examples. Others is complement fixation test, precipitation, and neutralization tests. And this one we'll look at them tomorrow. Then tertiary, you can also measure the antigen-antibody interactions in vivo, that is in the body. Okay, so these are some of the serological reactions that you can expect to see or to meet as you continue with this practice of serological tests. Now, some of the common terms that you will find: validity, which is a serological test that should provide an indication of which individuals actually have the disease and which do not. In other words, your test should be valid if it can give you the right information or the right diagnosis as pertains to the patient investigation. If you are looking at somebody who is infected with this particular disease, then your kit should be able to be valid. And this can be tested by using the previous samples that are known to be positive for this particular disease and the negative, and you have a new kit or a new reagent, then you can use the previous samples to run it before even you run on patient samples. And this is the usual practice, because sometimes any kit that comes, you don't know whether it is working for your population. Especially nowadays, people are trying to get more samples from every part of the world when they are doing their sensitivity and specificity test. But initially, you'll find like most were done on Caucasian and not from the African setup, and some kits were giving some different results. So your kit or your test has to be valid in terms of it has to identify what is negative and what is positive. And now let's look to this related terminology. Specificity is the ability of a test to identify correctly those who do not have the disease. In other words, the true negatives. So your kit has to have high specificity. In other words, it has to be true to its detection. If people are negative, the kit must report them as negative. And then there is also sensitivity, the ability of a test to identify correctly those who have the disease. In other words, the positive tests. So a kit that is good in terms of validity must have these two components: highly specific and highly sensitive. And for the application, I don't know whether some of you have been to this blood transfusion screening. Usually they use very highly sensitive tests when they screen, simply because anything that is closer to positive, it peaks. These kits may not be highly specific, but they're highly sensitive, meaning because you don't want to take chances because you are transfusing somebody. So you rather pick anything that even if it's not correct, so that you do more investigation. So that's what I'm trying to say. Like for these screening tests that are used, especially in blood bank, they're very sensitive, but not forgetting that they should also be highly specific. Now, if you want to do the calculation, I'm just giving you an example. We have talked about the sensitivity as the true positives, okay? And the specificity as true negatives. Now, if you have, for example, 100 HIV and AIDS cases, you run them, you find like 99 out of 100 are testing positive, and only one is testing negative. Now, what is your sensitivity? It is 99 because these are 99 that are positive according to your test over the total number of patients you tested. That is 100 times 100. So your kit gives you what? 99% sensitivity. And then there is also specificity. If you have, for example, 200, not 200, 199 positive cases, and then there's 9,701 cases who are negative, then you can still calculate your specificity from that formula that is provided. So for any kit, especially these serological tests or kits, they need to be highly sensitive as well as highly positive. And that's why whenever you see people are doing HIV testing, for example, they are using 2 rapid kits. And for whatever reason, somebody who is in early or phase of infection who has not yet seroconverted or produced antibodies, there might be a borderline results. One kit or one HIV rapid test might show positive, that's a weak positive, and another one negative. So what are you supposed to do? You can test the same sample using ELISA kit, because ELISA is highly specific and highly sensitive. And that's why people will confirm whenever there's discordant HIV results or any other investigation, you confirm with ELISA kits, because ELISA kits are more specific than rapid kits. Now, more terms, we talk about the quantitative test, which is measures the amount of antigen or antibody in the body. So when you talk about the quantitative, sometimes you want to look at the concentrations. Now, at what concentration do you expect to see a reaction, or beyond which there is no reaction? So some of these involves the dilutions when we talk about quantitative tests. But qualitative test, this is just to detect the presence or absence of an antigen. For example, in endemic infections, whereby people are exposed to, like, for example, say salmonella or the typhoid infections, getting positive and negative does not make any sense. But you have to find the titer of this particular patient. So if the titer goes beyond a certain limit, then it is considered as an infection case. So in that case, and that's why we said earlier on that you must choose your test properly, knowing the application of your test. Should you go in for quantitative test at what time? Should you go for qualitative test? Okay, and that is what the difference is. So quantitative test, you are measuring the amount of antigen or an antibody, while the qualitative is just a mere detection, whether there is presence of an antigen or not. And it can either way, be it an antigen or an antibody. Now, seroconversion is the development of a detectable specific antibody to a microorganism in serum as a result of infection or immunization. As I said earlier on, when somebody is showing seroconversion or you can detect the antibodies, it does not mean this person is infected. It could be he's vaccinated for the particular disease and is producing antibodies because of the issue of the vaccine. So in that case, it is just showing you somebody is having an immune response to an antigen. And for that reason, if you are looking for an infection, you must employ additional tests. For example, you can go for molecular techniques where you're looking at the antigens, maybe in acute infections where the antibodies may not be that pronounced. Then seroconversion, it is the opposite of seroconversion. This is when the test, seroreversion is when the test can no longer detect an antibody or an antigen in the patient's serum. So these are some of the terminologies that can apply to serological reactions. So usually in a hospital, they can always have these two, the qualitative and the quantitative tests. Now, in our usual infection curve, let's look at what happens. You have somebody who has been exposed to an antigen, and what you expect is that there will be production of antibodies. Specifically, the first antibody produced is IgM, which wanes over time, and it is overtaken by immunoglobulin G, which I said that is produced and it can stay for a long period, and that constitutes the memory. So looking at acute infections, the most important critical is to look for the presence of IgM. But when you are looking at the chronic infection, you cannot expect to pick the IgM, but rather you pick IgG. But in the case of an infection, like I've said, then you can expect an increase of the IgM, which can sometimes be like fourfold rise in case of an infection. And in that case, you have to request for the measurement of IgM if you know this is a particular disease. Maybe depending on the geographical location, there are areas that are known for reinfections over time. And these are some of the changes that you can expect. So by knowing the history from the patient, this is what the doctors rely on to request for a particular test. Now, in the laboratory, how do you get the serum? In other words, we are asking, what is serum? Serum is blood that is without the cells. You collect blood on a red tube, which doesn't have any coagulant, and you are allowed to settle. Then what happens, the clotting will take place, and now there will be centrifugation of the same sample. Then you can see, like what you see there, you get two layers, which is clearly indicated. There is the plasma, and there is the buffy coat, and there is erythrocytes. So this is a typical of coagulate the EDTA, which carries the clotting factors. And when you find like this one is done centrifugation, you can have these three layers of blood. That is the plasma, the buffy coat, and the erythrocytes. So we talk this as anticoagulated blood because it has certain chemicals, calcium, which blocks the blood from clotting. So this one, you can have it like EDTA. These are just the purple tops. But in the case of serum, we talk about the red tops, which doesn't have any anticoagulant within it. So the clotting factors takes it's their responsibility, and you find the blood does the clotting. And when you centrifuge down there, you will see there is the disintegrated red blood cells because of the centrifugation, and then you have the upper liquid, which is serum. So serum does not contain any anticoagulant. So it's a clear solution, as you can see. And this serum is very, very important in serological reactions. Sometimes we can still use the plasma as part of the serological functions. There's not much difference, but some tests require specifically you use serum, while others you can use both. So get to know the difference between plasma and serum. Because plasma contains what? Fibrinogen, but serum does not contain fibrinogen because it has been utilized in clotting. Now, serum preservation, you can get your serum and use the serum collection or storage vials. These are small tubes or microtubes. They range between 1 to 2 mLs. You can store them once you transfer from the original tube and later freeze. Now you can store them frozen, either within the fridge at 4 degrees or within -20 And for long term, you can even store them at -8. Sometimes people are asked, what is the use of the tube? So whenever you visit any lab, just get to ask, where do you store your serum? Can I see the tubes, the microtiters plates that are used for serological reactions and so forth. So you can be able to see all these reagents whenever you visit. And then for pipetting purposes, for specific volumes, you need serological pipettes, microtiter tubes, slides, and disinfectant jars or containers for that reason. As I said earlier, you must wear the gloves, and then use gloves must be disinfected or disposed properly. Now, important notes in serology: serum must not be hemolyzed or contaminated. So whenever you see your serum after you have centrifugated and seen it as separation between the debris and your serum, see that it doesn't have a red tint or showing hemolysis. And these are some of the things that you can request. Maybe whenever you are a lab, what's the difference between hemolyzed serum and non-hemolyzed serum? You need to make the difference, because if you use the hemolyzed one, you may not get the right results as supposed to be. So it's your responsibility to ask, how does this one happen? What causes hemolysis? And this has to do with even the time of collection. If somebody is puncturing several times, trying to get blood in difficult conditions, sometimes that causes hemolysis. So there are many things that can interfere, and that will affect, obviously, your results. Then check the expiration dates of the kits, diluents, and reagents. These are obvious things that need to be looked at in terms of the quality control. Kits, even though are tested against whichever disease or pathogen, must be considered biological hazards. Even though some tests show you this somebody didn't have HIV for that reason, doesn't mean it's free of other infections. So any sample has to be treated with a lot of caution. As I was saying, this is the micropipette, and you can see it is multi-channel micropipette, meaning you can pipette different volumes, at least 6 of them or 9, some are even 12 at once. And what you can see down there is the preparation or the dilution plates, which you can use. So these are multi-channel micropipette. So they can be used for different dilutions. Yeah, and what you see is that the plates can have different shapes. We have those that are U-shaped, there are those that are V-shaped, and there are others that are flat bottom. You can see them as you get exposed. Now, these are the last things that I want to talk about, the dilution. Now, why is it important to use dilutions in serological reactions? Because this is quite used in different lab procedures. And the reason is that it is important to dilute your samples, which can be critical if you're looking at especially quantitative, which I mentioned earlier on. Sometimes you want to look at the concentrations that gives you the best interpretation. And there are some techniques that rely on quantitative results, and there are others that rely on qualitative results. Now, let's talk about the dilution. Now, I said, whenever you get a sample, some of them are highly concentrated. Not only the samples, even the reagents. So the procedures requires that you do a little bit of dilution as per the manufacturer's instruction. It's not your instruction, you get the manufacturer's instruction. And the reason is that by you doing the dilution, you meet the requirement of that manufacturer. So the examples of the dilutions you are always requested, this is just an example. You can do 1 in 10, that is 1 to 10, or sometimes you can do more than that. Now, what is a dilution? You have your serum volume. For example, I can pick 20 microliters, I can pick 5, I can pick 20. Then I need to know my total volume in terms of the diluent that I'm using. And the diluent, sometimes people commonly use normal saline. So this can be used to dilute the serum without interfering with the component that we are interested, whether it's an antigen or an antibody. Okay. So if you want to look at the total volume, then you must find the serum volume used plus the diluent. If, for example, I'm using 1 ml of the serum and I'm using 9 ml of the diluent, the total volume is 9 plus 1, that is 10 mLs. So it's important to know the total volume. And we are looking at this is everything is in solution. Your serum and your diluent is in solution. So if you take, for example, 1 and the 10, your ratio would be 1 to 11, not 1 to 10, because 10 plus 1 is what? Is 11. So sometimes when people deal with solutions, they make this mistake. When it says 1 to 10, somebody picks 1 and a 10. No, you just pick 1 and a 9 because they are all solutions in that way. So to find the final dilution, you take the dilution of the preceding tube times the serum volume over the total volume. This can be, I'll show you the figure that gives you what we are talking about. So serial dilutions, these are a multiplicative dilutions that you can have. For example, 1 to 10, 1 to 100, 1 to 1000, and so forth. It's just arranging your tubes, maybe 5 of them. To each tube, add 1 ml. But remember the original tube, you are picking like 8 or 10 mLs or 10 microliters and then 9 mLs. So there's no formula here. You must maintain the ratio because the volume can be changed depending on what is achievable, but the ratio can be maintained. So you can start with the first tube, then pick something from the first tube and transfer to the second tube, then pick something from the same volume, and that you do serial transfers. And that's what that figure says. If you look at through, you start with the first tube, which is 1 ml, but that tube could be having even 5 ml in it. But you are only picking 1 ml, and at the same time, the diluent is 1 ml. So you are having half dilution. So you pick 1 ml from the first tube, transfer to the second ml, again pick the 1 ml. Then the last tube, you pick 1 ml and you throw that one away. So you maintain 1 to 2 or half dilution. Then you can still have another one, 1 to 5 dilution, meaning you pick 1 ml and you add 4 mLs in all the tubes, and that becomes like this, the other one we said earlier on. So you can pick it, and remember you move from 5 to 10 to 5 times 5 is 25, 25 times 5 is 125, and so that is another dilution you can make. Then there's also the third one, 1 in 10. You can pick 1 ml, and then you have 9 ml of the diluent, and then you transfer 1 ml from one tube to the other, and finally the last one you throw it. So these are things that you will perform, but remember, this will be guided by the manufacturer instructions. When they say you dilute, you have to dilute, and there's always instructions on which volume to dilute. Okay. Then this is the same, what we talk about the tube dilution. You can have a tube instead of the plate, and you'll
Yeah. Doc, I think you can unmute yourself.
Hello, can you hear me?
Yes, we can hear you.
Yeah, so let me just see. Okay, I think I was there. So I was saying you can do this dilutions also in a tube. If you, for example, you have your anti-serum and you have your normal saline 0.5 ml. So you can transfer 0.5 ml from the first tube, then you mix the second tube again, transfer 0.5 ml, and the last one you discard or throw it away. So you are maintaining some level of diluting factor, and that is very, very critical with these dilutions. So in summary, this is what I'm talking about. You can use the fast dilution when you have equal volumes, like 50 microliters of your sample and 50 microliters of your diluent. Then you pick from the first tube, which is from a half, you transfer the second tube, that is the second dilution. You continue up to 6 dilution, then you would have reached 1 out of 64. Now, suppose from each tube you pipette out 20 microliters into a microplate and react it. If, for example, you are looking at agglutination, you might find at the dilution of 32, there is agglutination reaching there. Then at 64, you don't find agglutination. So you can talk about your reaction reacting up to 1 to 32 dilution, and the rest is not reacting, or it can only react 1 to 8, and that ends there. So these are some of the applications that you can find, especially in agglutination. You can we look at examples where this issue of dilution works. Then serial dilution, just as a recap, is a series of sequential dilutions that are performed to convert a dense solution into a more usable concentration. And that's why sometimes you may not be able to pipette all these, but by knowing the concentration or the dilution factor, you can even do the calculations and get your results. The process of stepwise dilution, in other definition, you can talk about the stepwise dilution of a solution with an associated dilution factor. So it is important to know the factor that you are using. I have said you can decide. You use 1 to 5, you can use 1 to 2, you can use 1 to 20. And as you proceed, know that actually you are doing the multiplication factor for that reason. In biology, this serial dilution is often associated with reducing the concentration of cells, maybe in a culture, to simplify the operation. So that's why it is very critical to get to understand this principle of dilution, because you will use it quite a lot if you be working maybe in a tissue culture lab where you have the cells that you get from after you harvest them from the patient, and you need to subdivide so that you perform immunological tests. You need to know the dilution factor you are using. Or sometimes you can even culture or grow these cells to multiply. And if they are overgrown, sometimes you need also to dilute them and perform other assays. So it's very critical. Then dilutions are also commonly performed to avoid having to pipette very small volumes, like 1 into 10. These are 1 to 10. These are small volumes. If I ask you to pipette 1 microliter, for sure, that is too little. But if I ask you to pipette 20 microliters, that is reasonable. 50 microliters, that is reasonable. So sometimes the accuracy is reduced when you have these small volumes, because there's a lot of interference and it's not very accurate. And if the pipette especially is not the one that is regularly calibrated and serviced, you can be assuming to pick 20, 10 microliters or 8, but actually you're picking like 4 or 2, and that has implication on your downstream assay. I think those are the issues regarding the addition. So the formula is given. If you want to calculate the factor, the formula is the volume of the sample used over the volume of the sample plus the volume of the diluent. So you can get your calculation there. For example, my dilution factor can be calculated as 1 ml, especially if I use the first tube, then I have 1 ml plus 9 ml. So my dilution factor becomes 1 to 10 or 10 minus 1 for that reason. So this can be deduced even from different tubes that are used, especially when it is in a series of tubes. Okay, so this is the same explanation I've talked about your dilution factor from the first, the second tube. You can practice this at your own time. It's not a very difficult. If you see down there, the total dilution of the tube, for example, the second tube, you know, the previous one is 10 minus 1 times 10 minus 1. You know, you're dealing with the second tube, then you come to 10 minus 2. So this is the last slide that summarizes what we have been talking about in terms of dilutions. So they will be different, but remember you are starting with the original sample, which you are picking 1 ml, and then you can do dilutions. So thank you so much. I think I can share this with you. And lastly, what you see is the applications of these dilutions, especially in performing a number of experiments, and the limitations are also provided. I think the time is far gone. Let me stop there. I think tomorrow we'll carry over from there. So thank you so much. Have a good day.